Hi, Holly. Thanks so much for joining me today. I appreciate it.
Hi, David. Thanks for inviting me.
Absolutely. There are so many topics we could discuss today. I'm very familiar with Sciex, but I just wanted to focus on a few of those for our first conversation and introduce you to the audience. I know that you studied at the University of Toronto studying biological and environmental processes of PFAS. I was curious, what inspired you to focus on this area in your early research?
So, ironically, my undergrad was something completely different. I actually studied forensic chemistry for my undergraduate studies. And so, PFAS was completely foreign to me, never heard of these chemicals. And it wasn't until I took a third year environmental analytical chemistry course, and really it was just for fun. I was looking for a course to fill my time in my final year. And that's where I met Scott Maybury because he was the professor teaching that course. And if you've ever had a chance to listen to Scott present or even take one of his course, you'll very quickly get just swept away by how excellent of a storyteller he is. His lectures are always full of chemical structures, and he likes to use them to tell stories of how they undergo different environmental processes. So things like transformation, absorption to sediments, bioaccumulation in wildlife. And I think the first time he mentioned PFAS was when he was talking about how these fluorotelomere alcohols, which we come to know as aphthos now in the literature, or FTOH. So these are volatile chemicals that are used to synthesize fluorinated polymers and surfactants that are used in our commercial products. But not only that, they can also be present in these products as residual byproducts from the incomplete synthesis procedures. And so he had talked about how these alcohols can transform in the atmosphere and also biologically, either through microbial degradation or through metabolism in animal studies to produce the perfluorocarboxylic acids or PFCAs that we hear so often now in the literature. And then really from then on, I was just hooked onto this whirlwind that we've come to know about PFAS.
That's awesome. Yeah, environmental contaminants are certainly a very interesting but also a pressing issue. Reading over your LinkedIn page and your resume, I was really impressed by how much experience you have in different areas working at the Ontario Ministry of the Environment, and then even at Sciex, you worked both on the R&D and the global technical marketing side. I'm curious to hear how all those different experiences really formed how you think about our space and your perspective in general.
A lot of these moves were motivated by personal desire for growth. That being said, I'm really grateful to the years I've spent at the ministry. That's where I learned the rigor of proper QA/QC when you are validating analytical methods. And also where I got a lot of hands-on experience with sample preparation and LCMS MS analysis. And also learning more about environmental contaminants like not only PFAS. I was initially hired at the ministry because of my graduate experience with PFAS, but I also got exposure to learn more about pesticides, nanophenols, and other really emerging class of contaminants that we see a lot in surface water and soil and sediments, personal care products, and pharmaceuticals. And I think for me, by then I've already been an LCMS user for several years, including the time that I've spent in grad school. So when I transitioned to Sciex, I think my experience as a customer from the other end really helped with bringing feedback and experience from the field to help drive product development during my R&D days and also shape the content that I now currently put out either in the form of technical notes or when I have to present at conferences or in webinars. And I think having been part of academia as a grad student, then working in the government, and now transitioning to industry, it has really helped me because, especially when I have to talk to customers with very different backgrounds, I think they value the knowledge that I bring to the discussion, especially on topics like method development and troubleshooting, because they can tell that I have lived through their pain in the past, or in some cases, I'm still dealing with them on a day to day basis, which hopefully makes me more relatable to their experiences.
That makes a lot of sense. I can't relate to that in terms of that I was previously in the people's shoes who I'm communicating with, but certainly in terms of how broad LCMS is and organomation's evaporation equipment being used in all different sorts of labs, being familiar with what they're doing and some of their challenges, I can certainly identify a lot with what you're saying. Short chain PFAS compounds like TFA are emerging as really serious analytical challenges. What makes these compounds particularly difficult, and how are labs adapting their methods?
That's $1 million question for a lot of vendors who are trying to come up with solutions specifically to address these challenges. These compounds are probably some of the toughest ones that I've had to work with since grad school. And I've been working in PFAS for a long time. So, trifluoric acetic acid or TFA and some of the other ultra short chain PFAS that we hear them kind of being collectively talked about together. They are, if you look at their chemical structure, they're very small. So, and they're very polar. So, if you try to analyze them with the typical routine panel of PFAS on a reverse phase column in LCMSMS, generally you'll see really ugly peak shapes or they just shoot right off the column. So, you'll see that they'll have really early retention time. And this is problematic because generally a lot of other co-extractables that you see from the matrix, so things like inorganic ions, things that can lead to a lot of matrix effects, they also kind of elute very early in the column. So when you have TFA and some of these other ultra short chain PFAS co-eluding together with these other interferences, you generally your data quality can suffer because of matrix effects. And one thing that we have worked very hard with on and also with our partner at Phenomenex is to really be do our due diligence in terms of testing a bunch of different columns to see which one can give us the best results for TFA analysis. And a lot of this work, I attribute a lot of credit to our partners at Phenomenex. We were able to develop a very robust ultra short chain method using one of their Phenomenex Luna Omega PSC18 column, and that column has shown so much flexibility in terms of how much we can push the gradient in order to optimize retention and separation. So I think column selection and really doing your due diligence in method development for the LC grading conditions, that's a key for dealing with some of the poor chromatography that you typically see for these compounds. But on top of that, another really big challenge is background contamination that we hear a lot about. These compounds are, especially for TFA and PFBA, everywhere. We see them in our consumables, so things like pipette tips, LC vials, they're in our solvents, they're in our LC system. And most people working with PFAS already know this issue. And it used to be that you only have to worry about PFOA. So that's the C8 perfluorocarboxylic acid, or maybe the 6,2 dipap. So that's another fluorinated phosphate ester. And then gradually you start seeing that contamination kind of shifting over to things like PFBA and like PFHXA. So those are the C4 and the C6 chemistry. So really reflecting the industry shift to some of these shorter chemistry. And now we're seeing the dominance of TFA not only in the samples that we're interested in, but just everything, all the tools, everything that we're using in the lab. So, really, how we've been adapting to these problems is just diligent screening. You, for any consumables and solvents that you're planning to use in your assay, it is almost a prerequisite to ensure that you are testing everything, to ensure that you are using the materials with either zero or at least the lowest levels of contamination possible. And this is a must even if the products that you purchase are claimed to be PFAS tested, because you don't know what the vendors or the suppliers are monitoring. They may be testing for PFAS, but are they testing for specifically the ones that you're looking at? And I would suspect a lot of them have not incorporated TFA in their testing yet. So that's something to be mindful of. And also, even after testing, sometimes you might have to find yourself pre-cleaning these materials. So, like rinsing your files, rinsing your pipette tips with methanol, or running the water that you plan to use for reagent preparation through SPE ahead of time. That could really help. All these tricks could really help reduce a lot of the background before you use them in your assay. Sorry, that was a very long and long-winded answer, but it really has been a challenge trying to tackle some of these issues with these compounds.
No, Holly, I think that was a great answer. I spoke on the podcast with Ed from MacMod, and the whole MacMod team did a poster at ASMS on sources of contamination working on PFAS detection. Part of that really focused on older labs who are trying to repurpose an older setup for doing PFAS work rather than if you were to start a lab today where you could buy all this PFAS free specific equipment where your chances of contamination might be a lot lower just because of what you procured for your lab. So that was just a very interesting conversation.
It is. And although ironically, I've also heard anecdotes from customers who, like, we know PFAS is in our LC system. And that's why it's a must now that you have to have a delay column to kind of delay any separation that's coming from the background from your LC system. But I've also heard from customers, those who've had their LCs for decades, um, just the amount of rinsing, the amount of solvent that has flushed through the system, sometimes they get cleaner background than a brand new LC. So it's kind of like a double-edged sword. Newer labs have the luxury of going for products that have now having claims of being PFAS tested and hopefully lower levels. But at the same time, you never know. I think sometimes years of flushing could also help do the right trick too.
Absolutely. I was glad you brought up Phenomenex. Certainly, column selection is critically important. In terms of developing LCMS MS methods for PFAS detection and complex food matrices, what are some of those big technical hurdles or approaches that chemists need to choose the right solutions, whether it be columns or their mobile phase chemistry?
So in food, the biggest hurdle is probably matrix interference, and it's very common in food. And they're challenging because they are hard to predict and identify, and they can be different depending on the different food type. And even, for example, if you're looking at a lettuce sample, from one sample to another sample, even if they're both lettuce, you can see completely different profiles of matrix interference. So I alluded to this earlier, but I can't stress the importance of good chromatography method development because this is so key to not only show that you have good retention for the target PFAS that you're looking at, but also to make sure that they are very well separated from any matrix interference that could be present. And so this starts, of course, making sure you have the right column selected for your assay, but playing around with your mobile phases and also tweaking your gradient, also key to improving the performance of your column. Like one example is methanol tends to give better signal for PFAS, although acetonitrile sometimes offer better separation. And we've seen this for separating PFOS and some of these bile acid interferences that we often see them colluding together in matrices like seafood and eggs. In addition to column selection, it's necessary to incorporate cleanup during your extraction using solid phase extraction with the right sorbent. Phenomenex also has an excellent line of cartridges designed for this purpose. It's really to make sure that you can pre-clean your extract and remove those interferences before you inject them onto the instrument. So these are some ways that you can develop upstream before you actually inject your sample onto the analysis. But on the other hand, you can also leverage other tools, especially on the instrument side, to help increase the specificity of your target analysis. High resolution mass spectrometry, or HRMS, it has been used very frequently to provide secondary confirmation of any positive detections that you see. And really, that specificity is unrivaled because you are using that instrument to zero in with a very narrow window to extract out the exact mass of your target analytes to the third or fourth decimal place. And really such an effective way to filter out any isobaric interferences that normally wouldn't be distinguished on a conventional triple quad quadruple mass spectrometer. Some of our collaborators, for example, have used our Sciex 7600 system to resolve out interferences from matrices like clams, eggs, and salmon. I do want to make a plug for the National Institutes of Standard and Technology, or NIST. They have created this really good database called the PFAS Interference List, or PIL. It's freely accessible online. It's on their website. It's a great resource for anyone analyzing for PFAS in food, and they are interested to see what interferences have been identified from others. Because it's open source, anyone can contribute their findings to this database. So it's a great tool for researchers to share amongst themselves what has already been identified out there and save some time if they happen to be monitoring the same analytes, same matrix, and they see a colluting interference and they don't know what it is. This is a great tool for you to check beforehand before you go down a rabbit hole to figure out what that is.
I'm so glad you mentioned that interference list because I've never heard of that, and I'll certainly happy to check it out. One topic I wanted to get into today that I'm not very familiar with at all, so I was hoping you could introduce it and talk about why it's so problematic in food safety is the topic of masked mycotoxins. I saw that you recently presented on that at the North American Chemical Residues Workshop.
Yeah, I mean, full disclaimer, that work, I did not personally generate the data for that work. I give a lot of credit to what I presented at that NACRW conference to my colleagues from the applications team, and also some of the data that I presented were from our collaborators. So I myself am also a novice to the mycotoxins world. I did quite a bit of reading to make sure that I sounded like I knew what I was talking about. But yeah, so mass mycotoxins, they are metabolites. Generally, when we are exposed to chemicals, something like drugs, our body will metabolize that chemical through a series of conjugation. The rule of thumb is to make them more polar so that they are less toxic and can be more easily excreted from our body. Sort of the same thing with mycotoxins, too. When we get exposed to these chemicals through ingestion, these mycotoxins would undergo metabolism to form these mass mycotoxins, which are really just conjugated forms of that parent mycotoxins. In contrast, the mass mycotoxins is not happening in the human body, it's happening in the plant through plant metabolism. This is really the plant's defense mechanism for detoxification against regular mycotoxins. And the problem is, even though these mass mycotoxins are less toxic, they can still be present. They can still contaminate our agricultural commodities. There have been quite a bit of studies reporting their conversion back to the toxic compounds, the toxic parent mycotoxins. When we ingest these crops, upon ingestion, our body does the job of metabolizing them back into the toxic form. So, for example, the fusarium toxins like zearalenol, nivalenol, T2, and HT2 toxins, their modified forms have been reported to be able to hydrolyze back to their more toxic parent form through gut metabolism. And another issue is that these compounds are kind of under the radar right now. So they're not regulated. That's problematic because we are not routinely screening them for food safety testing. In fact, we might not even have analytical standards for all these mass mycotoxins. So we really don't understand the full extent of our exposure to them and ultimately what kind of health risk we are putting ourselves through this exposure. And that's why at that conference, I kind of emphasized the need for developing sensitive and also selective methods for both the detection for what we already currently know that's out there, but also developing methods that can comprehensively profile all of the natural toxins that are present in food matrices.
Awesome. Yeah, I think you answered my next question there about sensitivity limits. How do you see advances in LCMS, MS instrumentation enabling laboratories to complete those workflows more effectively, specifically for mycotoxins?
Yeah, mycotoxin levels are challenging because they're regulated around the world right now. And depending on which region you are, some are being regulated through their local legislation. But for example, in Europe, the European Commission probably has the most stringent and a harmonized set of regulated limits for a number of different mycotoxin and food combinations. The Codex Alimentarius or Commission, or the Codex for short, that's generally a good place to look at what it sets the guidance and standards and maximum levels for around the world. So a lot of different countries actually use this reference to set their own legislation with regards to mycotoxins. The challenging thing with mycotoxins is that the LOQs are typically the LOQs that are required to meet these regulated levels are generally pretty low, especially if you're looking at foods that are intended for consumption by younger children, because this is the group that is the most susceptible to health risk associated with mycotoxin exposure. So we generally see those maximum levels being in the range of sub to low ppb, which is quite low. And this is really a need for us to push, for laboratories to push their sensitivity limits much lower to be able to detect some of these mycotoxins at such trace levels in some of these food matrices.
I know that LCMSMS is dear to both of our hearts in terms of where we spend a whole lot of our time and energy. And certainly it's the gold standard for food residue analysis. But I wanted to get your take on any scenarios where LCMS/MS might not be the best choice and what alternatives or complementary techniques might be used.
That's a good question, given that my whole life has been about LCMSMS. So I would say not so much that it won't be the best choice, but there are some applications where LCMSMS may not be the predominant choice, but just one of many techniques that you could use for characterization. Microplastics come to mind for one of these applications. This is an emerging topic of increasing concern. They're used in so many of our commercial products, and they have been found in oceans, in wildlife, in our food, and even in our own bodies. It's very worrisome. I don't think you can go through a day on LinkedIn or in social media without hearing microplastics, kind of similar to PFAS. And from what I know, what I've read so far is that microplastics analysis has traditionally relied on more optical based techniques like Fourier transform, infrared spectroscopy or FTIR, or Raman spectroscopy. And it's really to visually examine the particle size, the shape, and the number of particles. There's also a study using pyrolysis GCMS. This is where they would heat up the polymer material in the microplastic to produce a suite of degradation products that you can analyze using GCMS. As I'm reading and learning more about microplastics, I had initially struggled to figure out how LCMS can fit in with this analysis. It wasn't actually until I came across a paper that was just published this month. Excellent paper. It was published on the Science of the Total Environment Journal. And it actually featured our Sciex 7600 Xenotop system. This is one of our HRMS system that was launched a few years ago. And that team, which a lot of this work was based a team of Chinese scientists that were using the instrument to characterize atmospheric microplastics in dust samples. If you look at the paper, they had used LCMSMS to characterize these nanoscale fragments of microplastic particles that have decreased in size over time from abrasion, UV radiation, and chemical oxidation. And so it was really interesting to see how they were able to use LCMS to monitor these nanoscale fragments. And they also used it to look at it from a polymer characterization angle. So they would target the monomers that had broken out and also for any other decomposition products from these microplastics too. And I think another way that we can also use LCMS to analyze microplastics is through their role as chemical carriers. So, as we all know, microplastics, I almost envision them as sort of like these little plastic beads with a ton of surface to absorb environmental contaminants. And so, we can use LCMS to analyze the desorption behavior of chemicals that are interacting with these microplastic surface, really to assess the role of these microplastic particles as, are they sinks or can they act as transport carriers of environmental contaminants?
Yeah, that's not, again, not an area that I know a whole lot about, but I'd be very interested in taking a look at that paper you referenced. I know you and your team spend a whole lot of time helping customers optimize or improve their workflows. What are some of the most impactful improvements that labs can make, whether it's on sample prep, chromatographic separation, or data processing to really reduce turnaround times but improve accuracy as well?
A lot of it depends on what matrix you're looking at and also the type of compounds you're looking at. I'm an advocate of less is more. A lot of it came from my experience with working with PFAS, minimizing your sample prep. And of course, that will also depend if your instrument sensitivity allows for it. But if you have the sensitivity, direct injection or dilution can save you a ton of time, reduce the cost of consumables and solvents, and also reduce the risk of human error. And more importantly, help minimize contamination sources, which is especially critical when you're looking at such trace levels. Another idea is also automated sample preparation is becoming very popular. I have been seeing more promotion of these technologies in webinars and conferences. Automation of workflows that involve solvent extraction, SPE, sample dilution, and even using automation to prepare your standards and your sample extracts. This can help increase lab efficiency and giving the scientists back their time to focus on other critical work. And at the same time, automation, as we have seen, there's a lot of evidence in the data to show that it can help improve reproducibility in the data. And added benefit of having this extra time back for the scientists, I think it's a welcome in this day and age where we constantly have to update our learning and educate ourselves on new tool. So things like AI and machine learning, these are being increasingly adopted to streamline and expedite workflows like data processing. I see a lot of value actually in using these tools to train peak integration algorithms in our software and applying them for high throughput data processing. This could also help not only with integrate like peak integrations for quantitation, but also reviewing the data, reporting the data, and really kind of streamlining that process to hopefully minimize user intervention. And I think finally, one thing that might not be talked about enough is that sometimes I think we don't know what we have at our own disposal. If you look at the LCMS software that typically comes, and this goes not only for Sciex, but this goes with any mass spec vendor. If you look at the software that comes with your mass spec, they are embedded with so many functions and features that are designed to make our lives easier. I think a lot of people don't realize that. I mean, David, you mentioned earlier, I did work in software R&D for many years at Sciex before I transitioned to my current role. At least I know for our software, we can perform a lot of complex calculations without the need to export that data to third-party software for post-processing. Our software is built with automated workflows for managing the batch and the queue. We have automated workflows for optimizing compound parameters, also for data processing. And all of this can be streamlined into a workflow that really helps minimize user intervention to increase efficiency. And I encourage customers to read that user manual and those release notes that get buried in the C drive. Some like that will really pay off because sometimes you don't know if you'll run into a feature that would really help save a ton of time for you.
Just a side note, but one thing I'd like to see my staff take more advantage of because I've found it to be so personally helpful is not using AI necessarily to do your full data analysis, but even just the idea of we've been so used to dealing with these really cumbersome instruction manuals for all sorts of different types of products. The idea that you can now ask for specific instructions on how to complete a certain task, I'm hoping that makes some of these really technical tools so much easier for us all to use. Even something like Excel, where you can go really deep with Excel and do a whole lot of
Yes.
data work.
Yeah, I mean, Google AI Overview has been a friend, especially if you Google something like mycotoxin regulations around the world. It does such a really good job of just filtering out and really honing in on the information that you need. Obviously, I think you still need to do your due diligence to make sure that the information it reports back is correct. And I think to your point, using AI for something like peak integration, that's still in infancy for a lot of processing for a lot of companies that are trying to incorporate this into their software product. It still needs to go through the rigor of having trained scientists with experience to make sure that the algorithm is picking up the right peak, doing the proper integration. But for things like trying to filter out extraneous information and really reporting back what you need, I think this is where AI can really help with streamlining that efficiency and just saving a lot more time.
In closing, I'd love to hear about what's going on at Sciex that you're excited to tell people about, any previews you have of things that might be coming up, really anything you'd like to talk about.
Not specific products, but more so, areas of focus that we are starting to consider. I already talked about automation and other ways to boost lab efficiency, but I think sustainability and greener lab practices will be a dominant theme in the next decade in food residue testing. I recently attended NACRW and AOAC International, and both conferences had sessions dedicated to this topic. And the talks were all focused on automated technologies for sample preparation, miniaturization, so using something like micro SPE for reducing solvent waste. So I predict that future method development in food testing will focus on how to incorporate some of these newer technologies to improve existing methods. So that could be in the ways of reducing sample volume, reducing the amount of solvent waste that you are accumulating, but also making sure that even though you have these new technologies, you are still getting the same good data quality and that they are still compliant with the acceptance criteria that are currently outlined in our regulator protocols. And kind of circling back to chromatography, which is sort of the overall theme of this discussion, my recent experience with developing a method for the ultra short chain PFAS is really that you have to be creative with the tools that you currently have. Because we were analyzing for a very wide panel of PFAS of different sizes, which included the ultra short, the short, and the long chain PFAS, we actually ended up combining different columns with very different stationary phase chemistries. So one was a helic and one was a C18 to almost create this mixed mode environment to broaden the selectivity for separating such a wide panel of PFAS. And so sometimes you just have to be very agile and tinker with the existing tools that you have. And maybe you'll come up with a combined solution that could be used as really good feedback to R&D for column vendors to help drive future product development. And so we're very lucky. We have a very strong partnership with Phenomenex, and this is something that both companies will continue to focus on to see how we can evolve our products to really address the changing needs of the food testing market.
Holly, this has been wonderful. I hope you're willing to come back in the future. We need to talk about UV filters and veterinary drugs, and there's so much other interesting work going on that's connected to chromatography for us to discuss. So I really appreciate you coming on, and thanks for your time.
Yeah, thank you so much, David. This was a great discussion.